The title of my talk is Prostate Cancer Molecular Biology Made Simple. It's obviously difficult to cover all aspects of prostate cancer molecular biology within a 10-minute talk, but I'll try my best to illustrate a couple of areas of interest. So I'm a group leader at Barts Cancer Institute. I run a molecular biology lab there, and also an honorary consultant urologist at UCLA and Barts Health, where I undertake robotic prostatectomy for prostate cancer. So a brief sort of overview of the natural history of prostate cancer. I'm not going to go into too much detail on the clinical elements here, but just to say there are a number of areas in terms of the natural history and progression of prostate cancer where an understanding of the molecular biology is important. And there are three areas that I'm going to focus on today. The first area is surrounding hormone therapy, which is really central to the management of prostate cancer. In late-stage disease, use of chemotherapy, docetaxel chemotherapy, and also I'm going to touch on an area that's of importance in radiation therapy for prostate cancer. So just going to go back to basics to give you a brief overview of some of the important facets of understanding the molecular biology of prostate cancer. So as you know, what is a real key element to understanding molecular biology is an understanding of DNA, and DNA is contained within the nucleus of the cell, packed together within chromosomes, and it is packed in a superhelix structure, which was discovered by Watson, Crick, Rosalind Franklin, and Maurice Wilkins, and it was actually Franklin who didn't get the Nobel Prize, unfortunately, for this particular discovery, although she did most of the work in terms of the X-ray crystallography work in understanding the structure of the superhelix. And the DNA itself contains runs of nucleotide bases, and they are represented by 4 key letters: G, T, E, A, and C. And it's the combination of these letters, different orders, that put together in strings to generate proteins. So this is the genetic code, as it's seen here, and it's a degenerate code. So what that means is that there's different ways that these amino acids, these nucleotide sequences can be put together, but they result in the same peptide sequence. So how are these proteins produced? Well, they're produced by transcription and translation factors. So these are molecules which enter the nucleus and bind to bits of DNA and recruit other molecules that regulate the expression of genes by an important molecule called the RNA polymerase that converts DNA into RNA, which is an intermediary molecule that also contains these runs of nucleotide bases. And this RNA is a molecule called the ribosome, and what the ribosome does is recruits other bits of RNA called tRNAs that are complementary to the RNA sequence that's linked together with an amino acid encoded by the genetic code. And it's the recruitment of the tRNA and linking up a peptide chain that ultimately produces the protein, which is what actually does things within the cell. So everyone knows androgens are central to prostate cancer management and to prostate cancer biology. So androgens are produced predominantly by the testis under control of the hypothalamic pituitary gonadal axis. So GRNH from the hypothalamus acts on the pituitary, which produces LH and FSH for gonadotropins, which then act on testes, produce testosterone, which then acts on the prostate gland. And once the testosterone arrives in the prostate glands, it's imported and converted to its active form dihydrotestosterone by the enzyme 5-alpha reductase. It's familiar, as you know, in terms of the treatment of BPH. DHT binds to the androgen receptor, which is a nuclear hormone transcription factor. So a transcription factor is a molecule that regulates gene expression. So once testosterone is bound to the androgen receptor, it dimerizes and is imported into the nucleus, where it binds genes within what we call androgen response elements that regulates expression of the genes themselves. And these genes underlie important functions for the cancer cells, such as growth and survival of the cell, but also regulates expression of certain proteins. So the KLK3 gene, for example, is a gene that encodes in PSA, which, as you know, we use for management of prostate cancer. So androgen deprivation therapy in its current form, and the faculty earlier were talking about castration, was discovered by Huggins and Hodges in the US almost 60 years ago now. And what they found was that reduction of testosterone by castration or estrogen treatment in animal models resulted in regression of prostate. And exogenous testosterone in the West, we use LHRH agonists or antagonists as androgen deprivation to suppress androgens, and these act via direct or negative feedback loops. So cell growth occurs in the cell via a cell division cycle. And there are 4 key phases to the cell division cycle. So there's the G1 within the cell, the G2 phase, which is, contains several checkpoints, and then the mitosis phase, where the single cell splits into 2 daughter cells. And that's important because the way that occurs is arrangement of the two splitting cells in metaphase here, aligned by the mitotic spindle, which contains microtubules. And the reason that's important is that these microtubules can be targeted using drugs that we use in prostate cancer. So docetaxel is a drug that we use commonly in advanced disease that cross-links these microtubules, prevents cell division, and causes cell cycle arrest and apoptosis in the G2M phase. And as you know from the studies from Chartered, early studies in CRPC, that docetaxel has an impact on prostate cancer patient survival, and also other taxanes that are used, such as carboplatin, have a similar effect. So unfortunately, after a period of time of androgen deprivation, patients develop a phenomenon called castration-resistant disease, where prostate cancer progresses at castrate levels of testosterone. And there are a number of reasons why this might take place, and I've just highlighted 4 reasons that have been largely looked at. The androgen receptor remains key to understanding castration-resistant prostate cancer, and CRPC can occur through amplifications of the androgen receptor, which makes them more sensitive to low or absent testosterone levels. Alterations within co-regulators of the receptor that will result in an increased androgen receptor activity. AR-independent growth of the cell, expression of mutations, so that this results in spelling errors in the DNA and mutant androgen receptors that are potentially more sensitive, or splice variant androgen receptors where the receptor is truncated and active even when testosterone is absent. Another mechanism is crosstalk with other receptors such as tyrosine kinase receptors. So AR antagonists used clinically. The 2 AR antagonists, AR targeting agents that have been used and cyclotrone acetate. They both target the androgen receptor, and they have slightly different molecular structures. CPA, cyclotrone acetate, is a steroidal anti-androgen, as you can see here from its structure, resembles the cholesterol molecule and steroid hormone receptor, steroid hormone molecules. Glutamide is a non-steroidal anti-androgen that is synthetically produced at PC progression. Now we've progressed on to next generation anti-androgens such as enzalutamide, which have an effect both pre-chemotherapy and post-chemotherapy. I'm sure there'll be some discussions on that today. And there are further generation anti-androgens such as apalutamide and darolutamide, which are also used clinically. Target the androgen receptor function is by inhibiting production of extra-gonadal androgen biosynthesis, which tends to occur in the adrenal gland in patients with CRPC. And you can target this by inhibiting key molecules in the androgen biosynthesis pathway, specifically the CYP17 17 hydroxylase enzyme. And we use a drug called abiraterone, which is a very potent inhibitor of this particular enzyme, and blocks production of this of testosterone through the precursor molecules. And you can see here that abiraterone is effective, and this particular study was showed in non-CRPC over placebo. So the other point that I want to highlight today is the importance of DNA damage and understanding of DNA damage. So DNA damage affects DNA structure and function and can occur affecting single-strand or double-strand breaks. And DNA is a double-stranded structure in that superhelix within the cell. So single-strand breaks are repaired by nucleotide excision repair, and single-strand breaks typically occur by pairs such as oxidative DNA stress. And double-strand breaks, which is how radiation works, they cause double-strand breaks and non-cell death, are repaired by homologous recombination and non-homologous end joining. So why is that important? Well, everybody knows that if you add radiotherapy to androgen deprivation therapy, which for a long time was really the standard of care for patients with prostate cancer, you improve patient survival. But no one really knows. The reason is that castration downregulates genes that are involved in the DNA damage repair response. So that prevents the androgen receptor and associated molecules in being able to repair these double-strand radiotherapy to improve cell death. So what are the DNA repair mechanisms that are important here? So there are two key pathways that are involved in DNA repair. One is homologous, one is the BRCA pathway, and the other pathway is the PARP pathway. So if you have a mutation in your BRCA pathway, so this is described to be a nationality Jewish founder mutation, so BRCA1 mutation, BRCA2 mutation. What you find is that the cells are able to repair DNA damage in the PARP pathway. And similarly, if you have a situation where you have functional BRCA pathway, if you target the PARP pathway, for example, using PARP inhibitors, then the cells are able to repair these double-strand breaks. However, if you have a scenario here on the right where you have mutated BRCA and you treat the cells with a PARP inhibitor, the cells are unable to repair the double-strand DNA breaks, and this results in cell death. So why is this important in prostate cancer? So you might be aware that olaparib is a PARP inhibitor that's been used clinically in prostate cancer. And this particular study looked at patients with metastatic CRPC and found that in patients who've had mutations within the BRCA genes or other DNA damage repair-related genes, they were more responsive to the PARP inhibitors than the patients who were biomarker negative. In other words, have normal repair pathways. Gives you an example of clinical practice of microbiology prostate cancer. If you want further information, there's a book out by Chappell, Cato and Pan, in which we have a chapter where we've looked at some other important features, both of prostate and other cancers. So this book should be coming out fairly soon. Thank you very much, everybody.